All right, good night. And it's about some minutes past the 11th hour at night, 11 p.m. I'm here at 1524 LaVista Drive, Decatur, Georgia. Just returning a couple of days ago from Los Angeles, California, where I started some brief lectures on the COVID virus. There and then at Dormshire, adjacent to UCLA, 1 minute walk away from the David Geffen Medical Center and 5 minutes away from the Ronald Reagan Medical Center. We did a brief epidemiology study, the chronology of pandemics. Well, here's yours truly, Dolphy Gilpin here tonight, and we're going to continue, as we had promised, with some more in-depth analysis of this COVID virus. Well, first of all, let us say know that the COVID virus, the COVID-19 as it is called, because it started in December of 2019, is RNA in nature. It's an RNA virus, and for that reason, we're just going to do a brief study of DNA and RNA to differentiate DNA and RNA before we touch the actual virus characteristic. And so we have DNA here, and the DNA is have two strands, helical, which is 2 strands, and a nitrogen base of adenine, guanine, cytosine, and thymine. That's the DNA. And those are the 5 bases, nitrogen base. And we know that the RNA is one-stranded, it's just one strand of the DNA, and it is relatively more unstable than the DNA. And so the RNA now, we have about 4 types of them. We have the tRNA, which is the transfer RNA, the mRNA, which is the messenger RNA, and the rRNA, which is the ribosomal RNA. And they have a nitrogen base of adenine as well, guanine, cytosine, but instead of thymine now we have uracil in the case of the RNA. And as I've said, we're doing a brief backwards of RNA since the virus is RNA. And we see that the ribonucleic acid, RNA, is a linear molecule, and RNA is made up of molecules, about 4 types of smaller molecules called ribonucleoside. And your bases are adenine, cytosine, guanine, and uracil, as we have said. And that the 3 types of RNA are the mRNA, the messenger RNA, the tRNA, transfer RNA, and the rRNA, which is ribosomal RNA. That RNA is unstable compared to DNA, and that the most stable RNA is the rRNA, which is ribosomal RNA. And we said, no, like the DNA, the RNA can store and replicate genetic information. Some on RNA, we say, no, that RNA viruses have very high mutation rates, up to millions of times higher than their hosts. And these high rates are correlated with enhanced virulence and evolvability, a trait considered beneficial for the viruses. Now, a small increase in mutation rates in the RNA viruses can cause RNA viruses to go genetically extinct. And this might be probably a factor in which we can increase the mutation rate in viruses and cause them to go extinct. This might be quite beneficial to us when we're trying to delete certain destructive virus. Now, many mutations cause organisms to leave fewer descendants over time. And the deleterious mutations are thought to have always outnumbered the beneficial mutation, and that this remain true whether the organism have a high mutation rate or a low mutation rate. All right, so we have discussed the basic knowledge for RNA, since we are going to be delving into this RNA COVID virus. And the last thing now, we're going to see that mRNA, messenger RNA, is a simple strand DNA, and it's the blueprint for the mRNA, which is transcribed from that DNA strand. So let's turn over now and continue. All right, so let's continue here. Okay, as we were saying now, that the COVID-19 virus, which is also named SARS for severe acute respiratory syndrome, and SARS-CoV-2, which is severe acute respiratory syndrome coronavirus of the second generation. And we know that the nCoV2 means it's a novel. It's a novel virus, meaning that it's new. So we have drawn this to represent our COVID virus 19. And it's RNA carrying the genetic instruction. Now, typical facts on this COVID virus 19 is that without preventative or protective measures, we can have exponential growth. And as we represent this sort of exponential curve here, to represent exponential growth, and this is without preventative measures taking place. And this sort of flattened out exponential curve here, which is when you have healthcare management system, we need to flatten the curve. This is desirous for healthcare management, to flatten the curve for this exponential growth, which would otherwise be without preventative measure. Now, this COVID virus have a spreading factor of about 2.6 times. We mark it as 2.6x. And say if we start out with 5 people in the first cycle, this exponential rate would be 18 in the second cycle, 52 in the third cycle, 140 people would be infected in the fourth cycle, and 300 people in the fifth cycle, because of this 2.6 rate of spreading, called the spreading factor. Now the virus, as we have drawn it here with these little dots here, and groove and dots, it invades the lungs and cause fibrosis and scarring. Now the manner in which it invades the lung is that these rounded things here, like the corona, these rounded part, interlock with these ducts here in the cells and tissue of the lungs, and they lock in, and then the virus invades, replicate, and propagate. And you notice I've drawn these things, and it's like this groove, it fits directly into this groove, like it docks, and it interlocks, and then it invades, replicate, and propagate in the lungs. and cause fibrosis or scarring. Another hypothesis is that the CoV, or the novel CoV2 virus of the second generation, hijack the ACE receptors and enter into the cells of the lungs, causing fibrosis and scarring, causing severe respiratory conditions. At first, it would have been a mild to moderate, and then the symptom would deteriorate into severe and acute symptoms, and then which will lead to progressive acute, leading to death if it's not stopped or checked. Now, our purpose as healthcare professional would have been to reverse the order from severe acute to mild to moderate and then to normality. That is, if the preventative steps was not breached. So once again, as we sort of review this little part here, we know that the virus is RNA in nature, and that means it's carrying genetic information, and it's RNA. And because of the instability in RNA relative to DNA, this mutative factors, as they say, for the RNA is about 1 million times more for certain RNA, and that we have 3 types of RNA: the mRNA, the tRNA, the transfer RNA, and the rRNA, which is ribosomal RNA, which is ribonucleic acid. All right, let us continue. Once again, we see now that the virus hijack the ACE receptors, invade the lungs, killing the natural killer cells and the T cells. And as a result, there's a depletion in the number of NKC, which is the number of natural killer cells, and the T cells, which are the immune system protective devices or components. And these NKC and T cells are specialized WBC, or white blood cells, and lymphocytes. Specialized white blood cells and lymphocytes. But what happens is that this virus invades, hijack the ACE receptors, invade the lungs, and kill these natural killer cells and the T cells, and cause a depletion, which causes the immune system to be compromised in such a way that the body has no defense against this virus. And here's another illustration of the virus here, which is a little corona, as they call it, hence coronavirus. Now, some researchers are now saying that the CoV2, or the novel CoV2, can stay within the body for about 30 days. And that the first symptoms are fever and dry cough, hypertension, and with pre-existing condition and the potential for rapid deterioration condition to SARS is more likely, which is a serious acute respiratory syndrome condition. Now, the first-line preventative measures can be realized by using soap and water and washing frequently. That is what the preventative measures have been taken here in our world today. The washing with soap and sterilization thing, and hand soap and detergents, and or various form of alcohol with 60% alcohol, and so on. And what this soap does is that it cracks the envelope of the virus and kill the virus. So that this is very effective if we continue to wash quite often. However, if the virus does infect and enter into the cell and enter into the lungs, as it's known to attack the lungs, then the preventative measure is breach or compromise, and it's no longer effective. So we have to go to what we call now treatment and cure. All right, so in the development of an antiviral, antibiotics or vaccine to fight this deadly virus, we know that one, as we said about the characteristic of the virus, is that in the mutation, if there's an increase in mutation, sometimes it causes the virus to go extinct. But what we, as researchers, can do, if we develop a vaccine or antibiotics that can destroy these, the structure of these, the virus itself, destroying it so that it can't dock with these locks here, the docking, and then invade the cell, then we could at least prevent the virus from invading the ACE receptors by destroying the sort of coronal here and the docks. Another way is to completely destroy the virus itself, and by so doing, we will prevent it from entering into the body and into the cells and the lungs and cause the damage. But most of the time, sometimes when we try to kill these microorganisms, as in cancer, we also destroy healthy tissues and cells, and this can be of detriment to the body as well if we continue to destroy healthy tissues. So that a more effective way probably would be just to destroy the structure, or the outer structure of the virus, and prevent it from invading the cell. So we have to develop antibiotics or vaccine that can have that characteristic property of destroying or altering the structure of the virus, or in other cases, increasing the mutation so that the virus can go extinct. So, all right, we look at the microbiology and some sort of pathology of this virus. Next, we will come and look at what we're doing in terms of treatments and cure and so forth for the present condition of the virus. And we know that this virus is deadly, it's lethal, it's devastating, this is a pandemic, and it has potential to do great damage to our world and to our nation.